So plant cell seen under a low power of a light microscope. Plant cell seen under a light microscope. At low power, we will only see cell wall, always draw two lines to show the thickness. Okay, and you will see a nucleus. That's it. So these are the structures that you can see under a low power of a light microscope. These joining gaps should not be there. You see, these are the gaps here. Joining gaps. So you will have some marks lost if there are joining gaps in your diagram. Okay, like this. So it should be continuous, smooth, sharp, non-feathery, unbroken lines with soft pencil. So when you draw this for your paper 3, you should make a note of it that there are some marks for quality of a diagram. Okay. A plant cell seen under high power of a light microscope. Okay. If we view it at high power of a light microscope, we will see a cell wall which is drawn with the two lines. Okay. This shows the thickness of the cell wall. If there is another cell associated with it, so you should draw another line here. For example, this is the middle line, and if there is another cell, okay, so this is another cell. Okay, so I have made actually the two cells, two adjacent cells. If there is another cell up, here, you can draw that like this. Do you see there are two lines here that show the cell wall of a cell, and do you see there are three lines here or three lines here? Why there are three lines here? Three lines where two cells are two adjacent cells. Are shown together. This is important to know when you make diagrams in paper 3. Then you can see there are narrow passageways in the cell wall between the two cells. These are known as plasmodesmata. Plasmodesmata can be defined as the two adjacent cells. Plasmodesmata are for exchange of materials between adjacent cells. So this is plasmodesmata. If a cell membrane has to be drawn, then you should draw another line. But as cell membrane is not seen under a light microscope, even at high power, so neither the cell membrane is drawn nor it is labeled here. Okay. Then you have a large sap vacuole. This is sap vacuole. It is single membrane round vacuole, and the membrane is known as टोनोप्लास्ट. इसको हम कहेंगे टोनोप्लास्ट. Okay, we have cytoplasm here. We will see a nucleus here. We can see one or more nucleoli or nucleolus. We will also see chloroplast, but the details of the chloroplast will not be seen. Then mitochondria and Golgi bodies. These are the structures seen under light microscope at high power. So here we have a plant cell. So you see here we have 3 lines. Why we have 3 lines here? As there are 2 adjacent cells drawn at this point. तो जब 2 cells इकट्ठे हम draw करेंगे plant cells के अंदर, तो then we will draw 3 lines where 2 cells are joining together, and where there is a single cell, then we will draw 2 lines. तो यहाँ पे 2 lines हैं, यहाँ पे 3 lines हैं. So no plasmodesmata in low power. Plasmodesmata are in high power, and no cell membrane under a light microscope in any case. ना ही तो हम draw करेंगे उसको, ना ही हम उसको label करेंगे. Is it okay?
Sir, can you show the point below joining gaps?
Yes. Seen under an electron microscope. Okay. Under an electron microscope, similarly, we start with the cell wall. So this is the cell wall. Okay, so this is one line. We will draw another line. Okay, so this is one cell. If we have another cell adjacent to it, so we will draw another line here. Okay. The middle line here is known as middle lamella. And this is cell wall of the first cell, and the other is the cell wall of the first cell. You see there are two cells. So you get those cell walls actually at this area. Here then we have a cell membrane. So cell membrane, we draw another line. Okay. So this is cell membrane. The other cell will also have cell membrane here. So this is the cell membrane of one cell, and the other is the cell membrane of the other cell. At plasmodesmata, the cell membranes are joined with each other like this. So if I enlarge this part further, and if I can show you, so you see there is middle lamella in between. This is middle lamella. You have cell wall here and cell wall here. Okay. There is cell membrane on this side and on this side. Both of these are joined here. And similarly, it is here too. So there is no cell membrane in front of these pores of plasmodesmata. Plasmodesmata के सामने cell membrane नहीं है. Okay, so ये इसका cytoplasm है, इस cell का, and this is cytoplasm of the other cell. So there can be an exchange of materials from one cell to another cell. Okay, so this is structure of plasmodesmata. Then we have a large sap vacuole. This is sap vacuole. We have nucleus here, which you know is a double membrane bound. Structure. This is an inner membrane and an outer membrane is like So this is an outer membrane. This collectively known as, यानी ये अगर outer membrane है, this is inner membrane. So collectively it is known as nuclear envelope. Inside it, it has one or more nucleoli, and it also has chromatin and this is nuclear pores. The outer membrane is extended into a network of membrane bound sacs or tubes known as endoplasmic reticulum. So this is endoplasmic reticulum. This endoplasmic reticulum has granule-like structures attached on it. So that is why it is known as RER, यानी rough endoplasmic reticulum. And we also have membrane bound flattened sacs called Golgi body. Then we have oval shaped chloroplast. The details can also be seen in the chloroplast, and we have mitochondria. The details of the mitochondria can also be seen. Mitochondria are, however, smaller than the chloroplast. Other than that, we have ribosomes. So this is mitochondria, and this is chloroplast, and we have ribosomes here. So this is a plant cell seen under an electron microscope. Is it okay?